Science on surfaces. A bigger perspective on the small.
So welcome to this podcast, Science on Surfaces, a bigger perspective on the small. Here we zoom in on science that impact our everyday lives. So today we'll talk about biomaterials and implants. We will zoom in at the interface between the living and non-living, and we will talk about what happens there at the surface of these foreign materials when they interact with the physical body. And we will specifically talk about what makes some of these materials integrate into the body and become a permanent part of an individual, while other materials can easily be removed even after a longer period of time. So my name is Malin Edvardsson, and here with me in the studio I have Professor Bengt Kasemo. You are Professor of Physics at Chalmers University of Technology. You are also a member of the Royal Swedish Academy of Sciences and the Royal Swedish Academy of Engineering Sciences. You have a long experience working with biomaterial research, and particularly you were involved with a project which was initiated by Professor Brånemark, who is also referred to as the father of modern dental implantology. And this project resulted in the Brånemark concept for titanium implants, and your research group was specifically involved in, for example, analysis of the implant surfaces, the implant bone interface, and this work led to analytical procedures for quality and production control, a new procedure for sterile packaging, and also an increased understanding for the biomolecule absorption and cell proliferation on different titanium surfaces. And also I have with me in the studio Dr. Åsa Westling. So you are a senior R&D Scientist at Wellspect Health Care.
Yes.
Wellspect Health Care, you develop and produce medical devices. So for example, urinary catheters and bowel management products for persons who have some dysfunctionality in the bladder or bowel.
Yes.
So for example, after a spinal cord injury or so. And your background is in chemistry and polymer technology, and you have been working for many years with coatings of catheters and polymer biomaterials and materials that should be performing a function in the body.
Yes,
yeah.
For a short time.
Yeah. So both of you, welcome.
Thank you.
Okay, so generally in the context of biomaterials, and you have to correct me here, but I am picturing that we have these two different perspectives in terms of time. So we have like the temporary or the permanent interaction between the body and this foreign material and the living organism. So for example, either the material should stay in the body permanently and integrate and become a permanent part, so integrate with surrounding bone and tissue, or it should just be there temporary and perform a function and then be removed, which is the case with, for example, a catheter.
Yes.
And so both of you have this long experience working with these biomaterials, and I think you represent the two different perspectives here. So you, Bengt, you worked a lot with the tissue and osseointegration, and you represent the aspect then of wanting the material to integrate into the body. And you worked a lot with the functional products. So we have mentioned a few situations where we have biomaterials. We have the dental implants, we have the catheters, and then we could have stents or pacemakers, contact lenses. And so there are so many different types of materials and that are interfacing different types of tissue. So we have many different types of interfaces that we are talking about here. But so let's talk about in general what happens when a material interface a body at the interface there.
Okay, should I
yes.
start? Because already there, independent of whether you're looking for something temporary or permanently, there are again many different perspectives. For example, if you're going to implant a titanium implant, the first step is actually the surgery. And the surgery prepares the site of implantation by causing quite a lot of damage, inflammation and so on.
Right.
And that's where the titanium implant is positioned. And then a lot of processes occur. The first one is that water molecules from the bioliquid, the blood, if you want, absorbs on the surface, and then small molecules like peptides, proteins, and this is on the time scale of minutes, maybe hours, and then living cells arrive, and they settle down on the surface, and then the healing process starts, which is days to weeks. We cover a lot of time scales already with the dental implant.
Yeah, and I think about devices or materials that come into contact, maybe not in a wound, but on the skin or in the contact of mucosa. Then, of course, there will be other things happening. And, for instance, for our urinary catheters, the catheter surface comes well into contact with the inside of the urethra and the bladder. And there, of course, we don't want too much interaction, or rather we would like very little interaction, because it's going to be used for a very short time.
That's true. And so, what aspects are important? And of course, certain aspects will be important for the short-term product that you're talking about, and other aspects are important if we want to integrate. But if you think about the surface body interaction, what aspects would be important then in these two different cases? If we can start by you.
Yeah, of course, the very interface is important, as you say. What the tissue, what does it see of the foreign material? And in my case, we have a very slippery surface which contains very, very much water. So I'd say it's mostly water that it sees, and also some hydrophilic polymer, which is well tolerated. And of course, there are always chemical substances in the material that come into contact with the tissue. And then, of course, it's important that these substances which can be released don't have any negative effect on the tissue, for instance. And this is important for very short time contacts as well.
So you're referring to an aspect of biocompatibility. You haven't
Yes,
mentioned
I'd
the word,
say.
but it's
It is true. It's biocompatibility.
which is essentially the body tolerating the material.
Yes. And of course it has to have its function, the material, but also the biocompatibility being exposure to different substances which may have some kind of reaction of the body and tissue.
And so then, of course, biocompatibility is important also for an implant. But then additional functionality must be crucial. So
the healing in of the implant can be along this scale of tolerable. You might want, ideally, you want something that promotes the bone to grow, bone promoting materials. But it's not necessary. It may be enough to have a bone accepting material.
okay.
So it accepts the body repair, what would have happened even without the implant, to repair the bone. And then when it meets the implant, it ties into the surface, and the body accepts the material. What you don't want is something that increases inflammation and causes tissue damage or even release of the implant. So there is a whole scale there, and which part of the scale that you want, promoting or acceptable, also depends on the status of the tissue. So a patient in good health, for a patient of good health, it might be enough to have something that is bone accepting. But for a patient with poor bone status, and so you may want something that is bone promoting. And
so bone promoting.
Bone promoting may be that the actual surface of the implant helps form bone.
Okay.
In reality, this could be, for example, that bone cells come from the surgical damaged tissue, and they settle down on the surface and they form bone, although bone is not yet in contact with the implant. So bone cells move from the intact bone tissue to the surface and starts to create bone there. And then it's called bone promoting.
Okay.
And like a material like hydroxyapatite has in many studies been shown to have this capacity to promote bone formation. So even if you place a piece of hydroxyapatite in a muscle, you can get bone.
Okay. That's and how long will that take then?
We're talking about weeks.
Weeks.
And so for a long time, and still there is a lot of research devoted to using this to get some advantage in the clinic. But as far as I have followed this, there are a few cases you get that advantage to the bitter end. To the bitter end, bone accepting is enough before you can use the implant for dentures and so on. So there was a lot of hope that hydroxyapatite should promote the healing process, but as far as I know, the success is relatively limited.
Okay.
But I also think like surface topography and such things, so surface sharpness may have an influence as well.
It has both a direct and indirect influence. The topography is now, we're talking about the topography on different length scales. So topography on the length scale of proteins can affect proteins. So for example, a very flat surface versus a very rough surface on the protein size scale can influence if proteins denature or not. And then there is another length scale, and that's the roughness on the size of cells. So if you have something like cavities for cells that fit for cells, they will react differently than on a flat surface on the cell length scale and so on.
Okay.
So you're absolutely right. Roughness, topography is also important. That has been studied in many studies by making microfabricated surfaces and showing that you can align cells on ridges or grooves.
Oh, interesting.
So then could you have both? So you would have some structures that are protein size and some structures that are cell size.
Exactly. So you can have superimposed roughness scales that work together for the ultimate result.
I was actually thinking...
And actually one thing that there's a more trivial aspect of that, the roughness just after implantation can also help to stabilize the implant, so it sits to let the other processes occur, so it's not so mobile.
Yeah, that's good.
Sorry, you were going to say something.
No, I was thinking functionality short-term and long-term. So for example, in your case with the implant, it's important that it integrates, but then once it has integrated, will it then stay integrated? Is that...
That's what you hope, yes.
That's what you hope,
and
but it's not...
What I hope, after many years of working with Brånemark, 5 years ago I got 2 dental implants.
Okay.
and the dentist surgeon who did this work for me for a clinic in actually at Vasaplatz, and he said, I'm going to the surgery of one of the legendaries of the titanium implants. And he did a fantastic job. It's been working for it's 3 or 4 teeth here.
Okay.
It's been working perfectly, permanently.
So you tested your own research,
eventually. That was after, must have been 15 years, 20 years after the first encounter with Brånemark. So I had seen the results and I trusted.
Perfect.
That's good. Okay, so we talked about what's important in terms of functionality and usage and so on a bit. And then the next step would be to design the material so that it induces these responses that we are talking about? Like in the case of a catheter, it should be easy to use, for example, to insert and remove. And we talked about the topography of the implant surface that can make it actually integrate. And so we didn't really talk about then what happens when a material integrates and one that doesn't. I guess it's just the different responses that are induced.
Mm-hmm.
Is that the situation, or?
It is. It's like a causal connection between the first milliseconds. On the millisecond scale, water covers the surface. But water coverage is very different depending on the material. So depending on the material, you get a water film in less than microseconds. And this water film is what the proteins meet. And depending on whether the surface water film is very stable or very unstable, proteins will react differently. They may denature or not. And then when cells come, it makes, of course, a big difference if the proteins are denatured or not.
Yeah.
And they react differently. So there is and then eventually that affects the overall healing, tissue formation. So there is a causal connection between the first fractions of seconds to minutes to days to weeks. And it's not the same, in principle, it shouldn't be the same surface that is ideal for all these timescales.
Okay.
And I know you're thinking about later on so-called time programmable surfaces.
Exactly.
And this is the background for thinking about programming surfaces so they are optimal at each time point. But that's very difficult to make.
Advanced, yes.
Yes.
So basically what you are saying is that a contact lens or a catheter would never stick because you haven't initiated this initial response. It's on a different pathway, so to say, basically.
Basically, what Åsa was mentioning is that on those products there is a lot of water. And this brings me to the question of fouling, anti-fouling, protein-resistant surfaces and so on. And protein resistance, that is that nothing happens with the proteins, is very much connected with the water. And therefore, for example, a hydrogel, which is a film which contains 80 or more % water, and which is also very mobile, is not a place where water-soluble proteins would settle down and be very strongly bound. They would be very weakly bound and release again. And that could be perceived as a protein-resistant surface or a non-fouling surface.
Yeah, depends on the application of
Exactly.
the implant or the device. And for instance, in my case, where we have short-term contact, we don't want to have the interaction. We don't want to have biofilm formation or fouling or anything. And it doesn't happen so fast on our hydrophilic catheters. The type of catheters that we have is called intermittent catheters. So it's like inserted into the urethra and empty the urine through the catheter, and then you take it away. And the persons that use it use 5 or 6 catheters a day. And then there are also the other type of catheter that's called indwelling catheter, and then the catheter is placed through the urethra into the bladder and attached there, and it could stay there for several weeks actually. This is the type of catheter used in hospitals or like in elderly care or so on. And there is a big problem with biofilm formation, fouling, bacteria coming, infections coming. So it's a totally different scenario when the catheter is staying for a couple of weeks before it's changed, or even a couple of days. So then urinary tract infection is a very large problem for those that need that kind of device.
Okay, so it's important to be able to control, you mentioned protein absorption or protein interaction with the material surface. And so what characteristics of the surface determine how these molecules will behave when they meet? Because there are so many different properties of a material. I'm thinking the material itself, you have, I don't know, you mentioned structure. So what aspects of the surface is important to design what will happen or determine what will happen? Is it possible to say something in general?
It's possible to say something in general. I guess two of the most common words that appear in such discussions are hydrophilic versus hydrophobic. And a very hydrophilic surface, like I mentioned, hydrogel-like, which basically you want no material there except water, but you need some molecules that keep the water there. That's a very hydrophilic surface, and it's usually also a protein resistant for water-soluble proteins. And so that's something you can aim for, to have such a surface if you don't want anything to happen. Because if it happens that proteins adsorb denature, then you start a process which goes in the direction that Åsa was mentioning. Eventually other proteins settle, bacteria or cells settle, and you get fouling. It starts on the molecular scale, but it spreads to the cell size scale and even tissue size scale. So that's one general thing one can say. You work along the axis of hydrophilic versus hydrophobic. But the reality is much, much more sophisticated than that, because along that scale you also have different charges, you have different topographies, you have different chemistries. So you have different chemical bonds that are formed on the surface. And if you really want to optimize the surface for, say, non-fouling, non-protein settling, you need to take these other aspects into account also. It means that the termination of the molecular groups that you build the hydrogel from may be important as well.
Okay, so is it possible to know beforehand and say that we measure the hydrophilicity and we measure the topography and we know the surface chemistry and everything, we've characterized all these aspects, can we know then that this material will be biocompatible and functional in the way we want it to?
I'd say it's important to test.
It's important to test. Okay.
Yeah, and then of course you could have a hint based on previous experience and so on, and you have like a set of test methods that you know already are important for the clinical outcome of it. But eventually, I'd say to see it in the final stage before launching a product, you need often to test it in its real environment, like being in the body. But that's a long way. You have to do lots of tests before you come there, but eventually that's it.
I agree completely. After many years of working with implants and materials, I would say there is nothing like a predictive power of the established knowledge. And that may sound depressing, but it's not, because even if you have searched for predictive power, you have developed a lot of methods to make a large-scale sorting. You know that in this corner of multi-parameter space of hydrophilicity, charge, terminating chemical group, you know that here it seems to work better. But it's not predictive. So when you use these methods that you have developed and the knowledge that you have in your data banks, then you have to do testing on the real object, which is human in this case.
Mm. Mm.
So even making predictions from animal to human is very questionable.
Mm. Mm.
So even if you have gathered a lot of knowledge and understanding, you cannot fully use that knowledge to guide you. It's
No, you
it's
can,
a guide, but it's not.
It can guide you into which clinical experiments you need to make and reduce both animal studies and clinical experiments, but it doesn't exclude them.
No. But it's, of course, if you make a minor change compared to something that has been tested before, then of course you know so much that you won't need to make large clinical trials,
Right.
depending, of course, on the difference compared to previous.
When you said a minor change, what could that be then?
There could be like design changes or minor chemical changes where you have done all these biocompatibility tests and you have so much experience that you make a judgment that this will not have an impact on the function or on the toxicity or the biocompatibility. So that will have to be done from case to case. But very similar products don't always have to be tested in a clinical trial if you do have sufficient experience from very similar products.
An event I remember from my work or my students' work was that there in the early days of the company being formed, Nobel Pharma, as I will call them, predecessor of Nobel Biocare, there was an inventor and also with a commercial company in the US who said that you don't need to use commercially pure titanium, which was the material of choice for Brånemark, CP titanium, which meant that the impurity levels were extremely low. He said that you can equally well use alloyed titanium with 6% aluminum, 4% vanadium. Because they form the same oxide. So he argued it's a small change.
Yeah.
And it took half a PhD, or so-called licentiate exam for us, a person whose name is Mikael Ask, to show that it's not true. Actually, this alloy forms an oxide on the surface, which is not only titanium oxide, but also vanadium oxide and aluminum oxide. And also with very different roughness of topography.
Yeah.
But his statement that he made just at the conference in a few minutes needed 2 years to oppose,
Of course.
to prove wrong.
To say if you think it's a minor change, it appears not to be a minor change but a major change.
It was a major change on the surface. What that meant then biologically is a different story.
Yeah, exactly.
You have to be careful.
Yeah, for sure. So we talked about materials being tolerated by the body, and we talked about designing materials so that they will induce a certain response. And we mentioned that for an implant surface, you ideally would like that to stay over time. But then we have a situation where the material degrades over time and then is replaced by tissue. We haven't touched upon that yet. Could you say a few words about that situation? Is that common or?
It's not common, very common. You could say that certain sutures, when you repair a wound,
right? Yes. Yeah, exactly.
they are designed to have that property.
Yes.
To resorb on a time scale that matches the healing of the natural healing.
So they disappear.
They disappear, and there are implants, not dental implants as far as I know, but there are other implants in soft tissue and in vessels and so on, where you exploit something similar, that part of the implant disappears. But I think it's an area which will expand. And for example, if you go into the hybrid area where we touch upon tissue engineering, you could say that you could use an implant surface, and before it's implanted, put a tissue culture on the implant to form the tissue that where the implant site is bone, for example. Because then the implant and the material forms like a scaffold for the tissue cells. And when they start to grow and form new tissue, you wouldn't like the implant to stay.
No.
You would like it to resorb.
Yes.
And then you would use something different than titanium. But this is still on the research level, but I'm sure it will come someday.
Yeah. And also then you could, with the right polymer material, you could design the degradation speed depending on what you put into the polymer. And then of course you have to take into account what is formed when the polymer degrades, so that it's not very, like, for instance, acidic and so on, and it's not good for the tissue surroundings. So there are different aspects in that area as well.
And this relates a bit to the time programming, because you would have the body seeing different surfaces over time, basically.
Absolutely, it's one aspect of time programming. What I was thinking of the first time I used the word time programming was more for the dental implant that makes something that meets water optimally when water comes in microseconds, and then meets proteins. So some part of the implant is dissolved.
Just det.
So you have an optimal surface for proteins, and then later on an optimal surface for cells and so on. That's a different type of time programming.
Yeah, and that would then be both in terms of, would it be in terms of the topography and chemistry, or all aspects?
All aspects that are important,
Okay, so we talked about biocompatibility. You mentioned also toxicity. And we've talked about functionality. And there are quite complex processes that are going on here. So what can go wrong? I mean, there seems like so many things can go wrong.
I know from our catheters. Actually, I have to be a bit proud because my company was the first one to launch this type of hydrophilic intermittent catheter. And in 1983, our low-fric catheter came out. And then previous catheters were like a plastic tubing, and in order to have low friction and non-traumatic for the patient, you could just put on a bit of gel and insert it. And now this first catheter that we had, it was with already a coating on the plastic tubing. You just added a little bit of water and it became very slippery, low friction, easy to insert and so on. And everyone was very happy, and it's easy to insert, and then you drain the urine. And after a few minutes, when the urine is emptied, you withdraw it, and then it was not as slippery anymore. What has happened? It was like a bit of stuck when you withdraw it. And it appeared that it was an osmotic phenomenon, since like inside the urethra there is tissue and there are cells, and of course there are small particles being salt in the tissue. And then our coating being hydrophilic, lots of water, then the water started migrating from the surface of the coating of the catheter into the cells in order to like dilute the particles through osmosis. And what happened was that the very surface of the coating dried out.
Of course.
Yeah. And since the coating it was stuck to the tubing, then you withdraw it, it's stuck.
Okay.
So and this was solved by adding salt to the coating. So
of course.
Then we have a right osmolarity in the coating to avoid this, and then it became as slippery to take out than it was to insert. So this is a typical surface phenomena.
And this is something that you wouldn't have discovered in the lab, so to say.
No, we wouldn't, because all the tests showed it was very slippery, and it was dried out for a while, still slippery. But it was when in contact with the tissue itself.
Right.
Yeah.
Right.
Any other complications? I'm thinking, what would you do if the implant doesn't integrate, for example?
Well, you were asking about when things go wrong, and I think the whole history of implants is filled with examples where things also went wrong. Before Brånemark came with his concept called osseointegration, where it's important to remember that it's not only the implant, it's also the surgical procedure. So he introduced a different surgical procedure that was used before. So before, the failure or success rate was usually never more than 50% or 60 or something like that. And when he reported at the conference in Toronto in, I think it was 1983, I was there and gave a talk, presented his results of 95% to 97%, depending on the upper or lower cheekbone. People just didn't believe it because it was too high. But eventually there came reports from other clinics, one in Toronto, and they had to accept. And what was the difference? Was it that he used commercially pure titanium? No, it was not only that. He was also using very gentle surgery. While the common knowledge, I may be out on thin ice now because I'm not an orthopedic surgeon, but the general knowledge was that you drill and you thread with very high speed so that the tissue, the bone tissue, dies. So it's actually a mineral. And you use a glue, a resin, to glue the implant to the dead bone.
Okay.
Yeah.
While he was using gentle surgery and you have living bone.
Of course different.
So it's very, very different. So this is an example that shows how things can go wrong. And I think also in the early days of stents, that are devices that are placed inside coronary arteries in the heart to keep it open, to expand it. In the beginning, the success rate was also very low, and it had to do with the surface treatment of the stent and also the mechanical, because it's a spring-loaded device. So both the spring loading and the surface treatment of the stent and the surgical procedure with the balloon dilation played a role. So many cofactors that make it work or not work. But generally I would say, as an answer to your question, the whole history of implantology is filled with failures in the beginning.
So what are then the challenges along, I mean, if you go from the development phase to whatever, or research and development and through all the way through the end user, what different challenges do we have? Like big challenges, are there any big challenges along the way that are more important than others? Or
I'd say it depends very much on the application that you have, and that you design a surface and device in order to suit that specific application. And in my case with the urinary catheters, they are used by people who maybe have impaired hand function, and then it's also important that they can use the device themselves. It has to be rather easy to open, and the risk for contamination has to be lowered, so that the design of the device is made so it is easy to use for the user, so that there will be low risk for infections and so on. And then has to be comfortable to use. They use it for lifelong maybe, and low traumatic and so on, have to be safe in all aspects to use. And of course that will be very, very different issues depending on the application.
Of course.
I think also when you ask for big challenges, not only small challenges, one important aspect is to make various implants and implant systems, or maybe not even only implant systems, but more temporary implants, to make them work not only for relatively healthy patients, but for patients that are not so healthy, and the elderly generation and so on. So the demand on the implant system increases as the health status of the patient is worse and worse.
Yeah.
So I think that's a very big challenge. And another big challenge is as tissue engineering grows and we start to understand and control tissue engineering more and more, to integrate implants, degradable implants, with tissue engineering concepts. So in the case of titanium implants, for example, hopefully make the tissue not only grow as bone around the threaded screw, but actually replace it with a root, dental root tissue
Of course.
via tissue engineering, that type of challenges. So the whole area of blood vessels, when you get to very small blood vessels, fouling and protein absorption and malfunction of the artificial vessel fails. So to go to smaller and smaller vessels is also a big challenge.
Because I'm thinking also we're growing older and older, and we are replacing perhaps more and more parts, or the need for is increasing. And I mean, is there any tissue that we cannot replace? For example, brain.
That we cannot.
That we cannot replace.
Brain tissue
Brain
is
tissue,
so
of
far,
course, but everything else is.
But there are not in the area of implantology, but in the area of brain research. There is research with neural cells, because even neural cells grow.
They do.
Exactly. And there are experiments to repair lost brain functions at the animal level. But one day I'm sure some of these will succeed to where you can actually inject or implant neural cells and repair some brain function. And then implants may come in as important scaffolds or carriers of these brain tissue cultures.
Interesting, and the brain is also plastic and
it's very plastic.
Can learn and change and help itself. So very interesting area.
So this is for the future. Any other thoughts on the future of biomaterials?
It's interesting, maybe more like individually suited implants, like specially designed for the patient, and so. And then, of course, there are. If you look at the society, there are more elderly people coming, and of course, I'd say health economy is important to have an play here.
Implantable sensors, and also implantable sensors, either for diagnostics, but also for releasing drugs or other biochemicals. So sensor actuator systems. And that's an area that develops by itself. What type of sensors can you build to sense glucose, to actuate injection of insulin and so on? That's a famous example. But there are many, many such examples of biochemicals that you want to detect with a sensor and then use the information to do something, to actuate. And that will put other types of demands on implantable materials, that they are compatible with the sensors. Sensors may have many different materials on the surface, and also the actuators. So that's an own area for implantology in the future, to build such systems and provide the systems with the right materials.
And then also signals could be sent, like for a smartphone with the results, and could be sent to the doctor, and there will be like home care and giving advice
Exactly.
at home. So that will that will come and simplify for many people.
Some very routine analysis. You could at least anticipate that they were incorporated in the way you say. I go to the care for control of the so-called PK value. It's the coagulation value. If you have some disturbance in the coagulation system, blood clotting, and that's first you take a sample at the local care unit, and then it's sent by mail, and 2 days later I get on the mail, and they get also at the care station, they get the information. But it's a rather elaborate procedure compared to having a sensor that senses that continuously and sends the information via the phone to the PK unit or AK unit at the hospital.
So is this a development that is ongoing already?
It's ongoing, yeah.
Okay.
I was also thinking about another challenge being the antibiotic resistance of bacteria. That depending on the device, that is a big problem, and I think it has to be addressed in other ways than using antibiotics and so on. There are different ways to look at it, and for instance we have an early research program using where we look into graphene, which is very, very nano-thin slices, and it appears that if they're standing not horizontally but vertically, then they're so sharp that they can cut up the bacteria and kill them by physical cutting. So, and that's an interesting area to look into. Then hopefully there will come more of these alternative ways to treat bacterial infections.
So then you could have that kind of coating, and then you wouldn't have the what you mentioned, the problem with handling and...
I'd say it would rather be when a device is in longer time in contact with the body, and when it comes to this biofilm and comes these bacteria and so on. Not for very short term contacts, but for longer. Yeah.
Okay.
A guillotine for bacteria.
Yes.
Right?
You could say that.
Guillotine.
That's true.
So I think that's what we had for this episode. It was a very interesting discussion. I learned a lot on the interface between a foreign material being in the body and how we can use our knowledge and understanding about those surface-related processes to design and steer functionality.
Yes.
So thank you for listening to this episode of Science on Surfaces, a bigger perspective on the small, with me, Malin Edvardsson, Bengt Kasemo, Professor of Physics at Chalmers University of Technology, and Åsa Westling, Senior R&D Scientist at Wellspect Health Care.
Thank you. Thank you.